Have you ever wondered why flowers are there? What are actual purpose of them? भगवान को फूल चढ़ाने के लिए or to present someone bouquet or flower? Absolutely not. Are these plants are doing something like charity to us? No. They are meant for sexual reproduction. तो फिर इतना fragrance और color क्यों है? इतना attractive color क्यों है? So yes, there is purpose for it. Not for us, and that purpose is not to serve us but to serve themselves only. So let's see what should be the purpose in chapter Sexual Reproduction Among Flowering Plants. Let's recollect what we have studied about flowers. A complete flower have 4 whorls: calyx, corolla, androecium and gynoecium, where each unit of calyx is called sepal, of corolla called petal, for androecium it is called stamen, and for gynoecium it is called carpel or pistil. Let's see what occurs prior to flowering. Flowering is predetermined process which begins with hormonal and structural changes leading to cell differentiation and forms floral primordium. This primordium determines location of flower much before they develop. Here in this diagram, what you see as dots are actually primordium. Out of all 4 whorls, androecium and gynoecium are sexual reproductive structures. So let's study about them one by one. If we collect stamens from different plants, we will find that they are of different sizes and shapes. Moreover, their arrangement in a flower also varies greatly. Stamens have 2 parts. Long and slender stalk is called filament, whose proximal end is attached to thalamus or petal, and a bilobe structure called anther. Each lobe is dithecous, containing 4 microsporangia, 2 in each lobe. Each microsporangia develop into pollen sac. Each pollen sac extends longitudinally through the length of anther and are packed with pollen grains. In the figure shown at right hand side is transverse section of an immature anther, where each microsporangia is surrounded by 4 layers. The outer 3 layer, epidermis, endothecium and middle layer, are protective in function, and during maturation they help in dehiscence of anther to release the pollen. Dehiscence means opening up of the anther. The innermost wall layer is called tapetum, possessing dense cytoplasm and generally possessing more than 1 nucleus, as they are undergoing cell division continuously. When the anther is immature, a group of homogeneous cells present inside the layer of tapetum called sporogenous tissue, which is present at the center of microsporangium. Each cell of sporogenous tissue is a meiocyte forming microspore tetrad. So each of these cells are called pollen mother cell, PMC, or microspore mother cell. As the anther develops, the cells of the sporogenous tissue undergo meiotic division to form microspore tetrads. Here cells of microspore tetrads are haploid, as only one set of chromosome is present. This whole process is called microsporogenesis. In fact, the meaning of word microsporogenesis is formation of microspores. Genesis means formation. This is TS of anther before maturation, and this is TS of anther after it matures and dehydrates. Each microspore formed from PMC develops into a pollen grain. Pollen grain, also called male gametophyte. Just recall gametophytic phase and sporophytic phase you have studied in chapter 3 of standard 11. Pollen grain is male gametophyte among angiospermic plants or flowering plants. In many flowers it seems to be yellow powdery substance, but color, size, shape and design varies among flower of different species. Pollen grains are generally spherical, measuring 25 to 50 micrometer in diameter. The cytoplasm of pollen grain is surrounded by cell membrane. Outside the cell membrane, there are 2 layers. The hard outer layer is called exine. It is made up of sporopollenin, one of the most resistant organic material known. It can withstand high temperatures, strong acids, and alkali. No enzyme that degrades sporopollenin is so far known. Although exine is not a continuous structure, so the aperture or gap present over there are called germ pores. The inner wall of the pollen grain is called intine. This layer is continuous and made up of cellulose and pectin. When the pollen grain is matured, it contains two cells, vegetative cells and generative cell. Vegetative cell is bigger, has abundant food reserve and a large irregularly shaped nucleus. The generative cell is small and float in the cytoplasm of the vegetative cell. It is spindle shaped, having dense cytoplasm and a nucleus. 60% of angiosperms shed pollen grain of two-celled stage, while 40% angiosperms shed pollen grain of three-celled stage. Two-cell stage is having a generative cell and a vegetative cell. Three-cell stage is having a generative cell and two male gametes, and these two male gametes are formed due to mitotic division of generative cell. Viability of pollen grain is very crucial for pollination. Before they lose their viability, they have to land on stigma. Viability of pollen grain depends on some factors like temperature and humidity. Viability of pollen grain varies among different plants, that is from 30 minutes in some cereals like rice, wheat, etc. to some months in some plants of Rosaceae, Leguminosae and Solanaceae family. Like sperms or semen of many animals, pollen grains of many species can be stored for years in liquid nitrogen, that is in minus 196 degrees Celsius temperature, called cryopreservation. Such stored pollen can be used as pollen banks similar to seed banks in crop breeding programs. Pollen grain as food supplements are available in two forms, tablets and syrups. They are rich in nutrients. Pollen consumption has been claimed to increase performances of athletes and racehorses. You might have heard about pollen in atmosphere causing allergy to some people. Yes, pollen grain too have some health issues. Pollen grains of many species causes severe allergies and bronchial afflictions, which often lead to chronic respiratory disorders like asthma, bronchitis, etc. Parthenium or carrot grass came in India as contaminant with imported wheat has become ubiquitous in occurrence. That means they are almost everywhere and causes pollen allergy. Pistil or carpels. These are female reproductive parts of flowers. Can you identify the flower shown in picture? It is flower of our very own saffron, kesar. In fact, we are eating carpels of these flowers. They are units of gynoecium. Gynoecium can be of two types, monocarpellary, multicarpellary. Monocarpellary are the one having only one carpel and multicarpellary are having many carpels. Again, multicarpellary are of two types, apocarpus and syncarpus. Apocarpus having separated carpels and syncarpus have fused carpels. Carpel or pistils are having three parts, stigma, style and ovary. Stigma is receptive part to receive pollen grain. Style is elongated slender tube in which pollen tube formation occurs. Ovary is basal bulged part and the cavity inside it is called ovarian cavity or locules. The placenta is located inside the ovarian cavity. You have studied about placentation, right? Axial placentation, marginal placentation, parietal placentation, etc. Megasporangia or ovules arise from these placenta. Number of ovules in an ovary may be 1, that is in wheat, paddy, mango, etc. to many, that is in papaya, watermelon, orchids, etc. Now we will study about megasporangia, which is also known as ovule. and it is present inside the ovary. This diagram is most important from this whole chapter. Let's understand the diagram. The stalk-like structure through which the whole ovule is attached to placenta of ovary is called funicle or funiculus. The region where funiculus or funicle fuses with ovule is called hilum. Ovule has two protective layers by which it is covered called integuments. There is a gap in the layer of both integuments called micropyle. Opposite to micropylar end is the chalaza representing basal part of the ovule. Integuments enclose mass of cells called nucellus. Nucellus cells have abundant reserve food material. Generally, an ovule has a single embryo sac located inside the nucellus. This embryo sac is nothing but female gametophyte. That means embryo sac is nothing but female gametophyte, as pollen grain is male gametophyte. This diagram is having much more information than you need. Here I have tried to do a doodle, and it is the first time that I am doing doodling. This one is embryo sac, in which at the chalazal end there are three antipodal cells, and at micropylar end egg cell to synergid cells are there, which constitutes egg apparatus. At the center of the embryo sac there is a cell called central cell. This is the only cell in embryo sac to be diploid. Remaining all other cells of embryo sac are haploid. Outside the embryo sac there is a region called nucellus, which is covering whole embryo sac. Then outside the nucellus there is inner integument, and outside the inner integument there is an outer integument. So there are two integuments, inner and outer. There is a region where both of these integuments are absent, and it is called micropyle. And the opposite end where these both integuments are present but they are fused with one another, that region is called chalaza, and that end is also called as chalazal end, where the antipodal cells are present within the embryo sac. And the region through which this whole microsporangium or ovule is connected to ovary is called micropyle. Megasporogenesis. The process of forming megaspores from megaspore mother cell MMC is called megasporogenesis. Again, the word itself means formation of megaspore. In an ovule, one cell from nucellus present towards micropylar end differentiates into megaspore mother cell. This megaspore mother cell MMC is a meiocyte, as PMC is a meiocyte, MMC is a meiocyte. So they undergo meiosis producing 2 cells and later 4 cells. From this megaspore tetrad, generally 3 cells will degenerate and only 1 cell will remain as functional megaspore. That's why this kind of development is called monosporic development. Likewise, there are bisporic and tetrasporic development too. But in 12th standard, we will be focusing only on monosporic development. Now these megaspore will develop into embryo sac. First, nucleus of functional megaspore divides mitotically to form 2 nuclei. Then each nuclei move towards opposite poles. 2 more sequential mitotic nuclear divisions result in formation of 4 nucleate and later the 8 nucleate stages of the embryo sac. Here, interestingly, cell wall formation is not followed by nuclear division, but after 8 nuclear stage, there is cell wall formation surrounding 3 cells at each poles. The remaining 2 nuclei present in large central cell situated slightly towards micropylar end. Group of 3 cells present towards micropylar end is called egg apparatus, consisting of 1 egg cell, that is a female gamete, and 2 synergid cells. These synergid cells have special cellular thickenings at the micropylar tip called filiform apparatus. These filiform apparatus guides male gametes of pollen grain for fertilization. Three cells lying at the chalazal end are called antipodal cells. Remember one thing, during description of embryo sac, we will never mention upper end or lower end, but micropylar end or chalazal end. Thus, a typical embryo sac at maturity is 8 nucleate but 7 celled. Pollination. We know what is pollination. It is simply transfer of pollen grain from anther to stigma. Pollination is of two types, self pollination and cross pollination. Again, self pollination is of two types, autogamy and geitonogamy. Here, pollination occurs within the same flower, hence, flowers must be bisexual. Yet, complete autogamy is rare phenomenon. As for autogamy, dehiscence of anther and receptivity in stigma should occur together. Moreover, they should lie close to each other. Some plants such as Viola, oxalis and Commelina produces two types of flower. First, cleistogamous flowers, underground flowers which never open at all. So there is no chance of pollination among two different flowers. As pollination occurs within the closed flower only, no pollinating agents are required even, and gradually seeds are produced there. Another type is chasmogamous flower, grows at aerial parts of plant above the ground with exposed anther and stigma. Here cleistogamy is really an advantageous in maintaining pure line of varieties. Geitonogamy is simply pollination between two different flowers of the same plant. Although it is functionally cross-pollination as pollinating agents are required here, pollination occurs within an individual and hence belong to self-pollination. Cross-pollination. Cross-pollination is also called xenogamy. Here pollination occurs between two different plants. Pollinating agents. Plant uses biotic factor or abiotic factor as pollinating agents. Only a small portion of plant uses abiotic agents. Most of them are using biotic agents, that is animals. So let's start with abiotic pollinating agents, that is wind and water. Pollination through wind or water is a chance factor. So there is loss of huge number of pollen grains. To compensate the loss, they produce large number of pollen grain than the number of ovules available for pollination. Among abiotic pollination, wind is very common. Here pollen grains are light in weight, non-sticky, so that they can be blown away with the wind. Flower possess well exposed stamens and large, often feathery stigma to easily trap airborne pollen grains. Here, often one ovule present in each ovary. In corn cob, the silky tassels you are seeing in the picture are nothing but stigma and style, which wave in wind to trap pollen grain. This kind of pollination is common among grasses. These are seeds transformed from ovules of flowers after pollination. Pollination by water is very rare in angiosperm and limited to about 30 genera, mostly monocotyledons. In contrast, among algae, bryophytes and pteridophytes, water is the only medium of transport for male gametes. It is also believed that distribution of bryophytes and pteridophytes is limited because of dependence of water in transporting male gametes and fertilization. Also, in most of the aquatic plants like water hyacinth and water lily, the flower emerge above the level of water and are pollinated by insects or winds. Anyways, examples of angiosperms in which water is pollinating agents are Vallisneria, Hydrilla in fresh water and several marine grasses as Zoostera. In Vallisneria, female flowers reach the surface of water by long stalk and whole male flowers or pollen grains are released onto the surface of water. As you can see in the picture, in which the female flower is surrounded by many tiny released male flowers. They carried away by water current passively. Here, sometimes pollen grains are ribbon like or even having mucilaginous coating for protection from getting wet. After pollination, female flower is drawn inside the water by getting spiral shape to the flower stalk. Flowers pollinated by wind or water are not very colorful, neither they have nectar. Sometimes we can't even identify them as flower, as we expect flower to look attractive and or possessing fragrance. These flowers have neither of them. Pollination by animals. Bees, butterflies, flies, wasps, beetles, ants, moths, birds like sunbirds and hummingbirds, and bats are common pollinating agents. Although among whole animal kingdom insects are major pollinators, large animals such as lemurs, arboreal rodents, reptiles like gecko lizard and garden lizard are reported as pollinators. Flowers pollinated by animals are having specific adaptation, that is large size, being colorful, having fragrance and possessing nectar. Even if flowers are small in size, they clustered into an inflorescence to make them conspicuous or easily visible. Animals are attracted by color and or fragrance of the flower. But for attracting flies and beetles, flower produces foal. To sustain visits of these animals, flower produce rewards in form of nectar and pollen for their nutrition. So animals visits flower to have these rewards and carries pollen grain from anther to stigma and achieves pollination. There are some special cases also like Amorphophallus flower, which is about 6 feet tall, provides safe place to lay eggs. A same type of relation exists between yucca plant and moth, where moth is laying eggs in the locule of the ovary and flowers get pollinated by the movements of the moth for laying eggs. Thus, life cycle of either of them cannot be completed without each other. For sexual reproduction, they are in obligatory, means compulsory. They are in obligatory relationship. One day, I asked my friend, he is a farmer, to cut a papaya plant which had never bare a fruit in his farm. He refused. He said that because of that papaya plant, other papaya plant bears fruit. How? You'll get the answer after studying the topic. Outbreeding device. Most of the plants are hermaphrodites, means bearing both female reproductive part and male reproductive part. So self-pollination can be done. But continued self-pollination results in inbreeding depression. Inbreeding depression means reduced ability to survive. So flowering plants have developed some devices to discourage self-pollination and encourage cross-pollination. First, that means first device, in some plants maturation of anther, release of pollen is not timely synchronized with receptivity of stigma. Either pollen is released before stigma becomes receptive or pollen released much after stigma becomes receptive. Second, position of stigma and anther are very farther from each other to reduce self-pollination. Both first and second device prevents autogamy. Third device. Some plants exhibit self-incompatibility. Self-incompatibility. That is, pollen grain from the same plant can't germinate over stigma. This is genetic mechanism prevents self-pollen to germinate on stigma or pollen tube growth in pistil. Fourth device. Bearing unisexual flowers means either androecium or gynoecium is absent in flowers. So here flowers are either male or female. Male are the one who does not bear gynoecium and male who does not bear androecium. Example, castor, maize, etc. This prevents autogamy. But what about geitonogamy? To prevent geitonogamy, Whole plant themselves are male or female. Means plant themselves are dioecious. Either a plant will have male flowers only or female flowers only, as in papaya. Male flowers does not have ovary, so they cannot be transformed into fruits. But their pollens are very crucial for the flowers of female plant, and these flowers transform into fruits. Pollination doesn't guarantee transfer of right type of pollen on stigma. Wrong type of pollen, that means pollen from same plant or pollen from different species, also falls on stigma. Stigma can recognize right type and wrong type of pollen. If it is right type, then and only post-pollination events leads to fertilization. Wrong type of pollen are just rejected from these events. In recognition of pollen grain, several chemical components of pollen grain and pistil interact with each other. Some of these components are identified recently by botanists. After recognition, right type or compatible pollen forms pollen tubes, comes out through germ pore. What is this germ pore? Gap in exine, in sporopollenin. The content of pollen grain move into pollen tube. It grows through stigma and style and reaches ovary. Here, if pollen grain is in two-celled stage, we have discussed earlier three-celled and two-celled pollen grain. So here, if pollen grain is in two-celled stage, generative cell will divide mitotically to form two male gametes. Here, first two pictures are images of pollen tube on stigma and through style respectively. Pollen tube after reaching ovary enters in a ovule through micropyle and specifically to any one of two synergid cells through filiform apparatus of synergid cells. So this whole event starts right from recognition of pollen grain till reaching of pollen tube inside the ovule is called pollen pistil interaction. Pollen recognition process is very significant for plant breeders to manipulate, especially in case of incompatible pollinations, to get desired hybrids. For producing hybrids, artificial hybridization is one of the major approach used by breeders for crop improvement programs. In this crossing experiment, only desired pollens are used for pollination and stigma are kept protected from unwanted pollens. This is achieved by two techniques, emasculation and bagging. First of all, anthers containing desired pollens are removed using forceps before they matures, before anther dehisces. This process is called emasculation. Emasculated flowers are covered by bag of suitable size made up of butter paper to prevent contamination by unwanted pollen. This technique is called bagging. When stigma becomes receptive inside the bag are taken out and mature pollen grain collected from anthers are dusted on the stigma. Then flowers are rebagged immediately. Lastly, fruits are allowed to develop. If the female parent produces unisexual flowers, there isn't any need of emasculation. Female flowers are bagged before they open, that is, they are bagged in their bud form. When they become receptive, pollination is carried out using desired pollen followed by rebagging. Double fertilization. Here diagram of embryo sac and progressing pollen tube are given. Just observe how pollen tube is progressing within the embryo sac and how two male gametes are released and these male gametes get fused with their respective cells. As you can see in the diagram, after entering of two male gametes through synergid cells, one male gamete fused with egg cell and other with secondary nucleus. The fusion of male gamete with egg cell is called syngamy, results in formation of zygote. The fusion of male gamete with secondary nucleus is called triple fusion, forming primary endospermic cell PEC. As PEC is triploid, results from fusion of haploid male gamete and diploid secondary nucleus. Here, fusion is occurring two times, one with egg cell and other with secondary nucleus. Hence, called double fertilization. Gradually, zygote develops into embryo and PEC, that is primary endospermic cell, also called as PEN, primary endospermic nucleus, develops into endosperm. And also, ovary develops into a fruit and ovule develops into a seed. All this development includes in post fertilization. Observe the picture carefully, especially labeling. What is fruit we will discuss later. First we will study development of endosperm. PEC, that is primary endospermic cell, divides mitotically repeatedly and develop into triploid endosperm tissue filled with reserve food material and are used for nutrition of developing embryo. In most common type of endosperm development, first primary endosperm nucleus, that is PEN, undergoes successive nuclear division producing many free nuclei. This stage of endosperm is called free nuclear endosperm. Subsequently, cell wall formation occurs and endosperm becomes cellular. So earlier, endosperm was having many nuclei in a cell. The number of free nuclei formed before cellularization varies greatly with different species. In a tender coconut, the coconut water is free nuclear endosperm, having many nuclei, also called liquid endosperm, and surrounding white kernel is cellular endosperm. Endosperm may either get consumed completely till seed matures or it may persist in mature seed. Remember this, huh? This will come again when we will study about seed. Endosperm may either get consumed completely till seed matures. Means when seed is matured, there is no endosperm. Endosperm is fully consumed. Or it may persist in mature seed. Means it will be available for nutrition during germination of the seed. That's why it is still persisting even after maturation of seed. Understood? So there are two types of seeds according to presence of endosperm in matured seed. We will come to that part later. You must have seen this kind of coconut, right? Have you ever wondered what this white colored structure is, which is present on the cellular endosperm, that is white kernel of coconut? It is embryo. Embryo development. Embryo develops at the micropylar end. As egg cell was situated at the place, most zygotes start divisions after certain development of endosperm. This is an adaptation so that nutrition gets available to dividing cells of developing embryo. Though seeds differ greatly, the early stages of embryo development are similar in both monocotyledons and dicotyledons. Development of embryo is called embryogeny. The figure shown here is of embryogeny of dicotyledonous seed. The zygote here give rise to proembryo and subsequently to the globular, heart-shaped and mature embryo subsequently. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. There are two different regions at either end of the embryonal axis. One end is called plumule, which will grow into shoot, and another end of embryonal axis is called radicle, which will grow into root. The portion of embryonal axis between cotyledon and plumule or shoot tip is called epicotyle. The region of embryonal axis between cotyledons and radicle or root tip is called hypocotyle. The developed root tip is covered with root cap. Embryos of monocotyledons possess only one cotyledon. In grass family, the cotyledon is called scutellum, situated at one side of the embryonal axis. At the lower end of embryonal axis has radicle and root cap is enclosed in an undifferentiated sheath called coleorhiza. The portion of embryonal axis above the level of attachment of scutellum is epicotyle. Epicotyle consists of shoot apex and few leaf primordia enclosed in a hollow foliar means leafy structure called coleoptile. Seed is the final product of sexual reproduction in angiosperm. It consists of seed coat, cotyledon, and embryonal axis. The cotyledons are simple, generally thick structure, swollen due to food storage. Mature seeds may be albuminous, that is endospermic, containing endosperm like castor, maize, wheat, barley, etc. All these seeds are having endosperm while they are matured. And another category is non-albuminous or non-endospermic, that does not contain endosperm after they get matured, like groundnut, pea, etc. On what basis a mature seed can have or lack endosperm? We have studied already, right? Occasionally, in some seeds, remnants, that is, remainings of nucellus, are persistent. This persistent nucellus can be found in seeds of black pepper, beetroot, etc., and is called perisperm. We know that dormancy is also a very important event in seeds. While maturation of seed, they are relatively dry, just 10 to 15% moisture by mass. Also, the general metabolic activity of the embryo had slowed down, which is actually called dormancy. Seed are covered with tough protective seed coats formed from integuments. Still, there is a persistent small pore in seed coat of seed which is called micropyle. Yes, it is the same micropyle which is present in the ovule. Here, it facilitates entry of oxygen and water to end dormancy. So, in favorable condition, seeds germinate. Pericarp. The term fruit is for layman, not for biologist. The biological term for it is pericarp. It is covering of seed developed from ovary and meant for dispersal of seed. Pericarp may be fleshy like in mango, orange, guava, etc. or they can be dry as in groundnut, pea pods in which pea seeds are arranged. You have seen the diagram of pea pod and pea seeds earlier. Pea pod itself is called pericarp or fruit. Here in pictures what you see as exocarp, mesocarp and endocarp are just different layers of pericarp. However, in some plants, thalamus also develops into fruit like in apple, cashew nut, strawberry, etc. Such type of fruits are false fruits and they are not pericarps, as pericarps or true fruits develop only from ovary. Here in picture what is labeled as endocarp, mesocarp are different layers of pericarp. So pericarp is there in apple core, not the part which we are eating. In spite of fruit formation is result of fertilization, some fruit forms without fertilization. For example, banana, grapes, etc. Their parthenocarpic fruits can be observed. Parthenocarpy means formation of fruit without fertilization. Parthenocarpy can be induced by applying growth hormones, plant growth hormones, like auxin. Yet, wild varieties of these plants produce seeded fruits. Now, let's see what are the advantages with the seeds. Processes like pollination and fertilization are independent of water, which are required for sexual reproduction in bryophytes and pteridophytes. So here, these processes does not depend on water media. Seeds have better strategies for dispersal to distant places using pericarp. In fact, sweet and tasty pericarps, as in mangoes and oranges, are for dispersal of seeds. Dispersal of seeds, right? Not disposal. Fruits are tasty, so arboreal animals dwelling on trees eat this fruit while traveling among trees, and seeds are thrown much farther from their mother plants. Third, young seedlings have well enough nutrient to nourish themselves during germination. Fourth, hard seed coat provides protection from mechanical injury. Fifth, as seeds are product of seed germination, they generate with new genetic combination leading to variations. Sixth, in agriculture seeds are used extensively. Seventh, dehydration and dormancy like features are very crucial for storage of seeds. In fact, some plants have extremely long seed dormancy, that is, they are viable to sow even after hundreds of years. Lupinus arcticus, shown at left hand side, their seeds found from Arctic tundra due to excavation having estimated age of about 10,000 years are viable. That is, the seeds can be sown, germinated, and plants produced will produce flowers. Also, during excavation of King's Harrod Palace, seeds of date palm was found which are viable even after 2000 years. Moreover, some plants have thousands of seeds in a fruit, as an orchid shown in left figure. Some plants produce billions of seeds in a plant bearing many fruits consisting of many seeds. For example, ficus, that is banyan plant, shown in right figure. Note that number of seeds in a fruit depend on number of ovules in ovary. Just check the size of seeds here. These seeds are of parasitic species Orobanche and Striga. They are of half millimeter in length. Now just consider all the aspects of seeds. You will understand how significance and dominance seed bearing plants are over the globe because of having seeds. Now next topic is Apomixes and Polyembryony. A few flowering plants such as some species of Asteraceae, which includes sunflower also, and grasses have evolved a special mechanism to produce seeds without fertilization called apomixes. Here, asexual reproduction is mimicking sexual reproduction, and there are several ways of development of apomictic seeds. That is, one of them is parthenocarpy. In some species, the diploid egg is formed without reduction division and develops into the embryo without fertilization. More often, in many citrus and mango varieties, some of nucellar cells surrounding the embryo sac start dividing, protrude into embryo sac, and develop into the embryos. In such species, each ovule contains many embryos, and this phenomenon is called polyembryony. Just observe the number of plants developing from a seed. Hybrid varieties for food and vegetable crops are extensively cultivated and they have increased productivity tremendously. But there is one problem with hybrid varieties also, that we have to produce hybrid varieties every year. We cannot sow the seed from the hybrid varieties and get the plant of same qualities, means of hybrid qualities, because the genes present in seeds are segregated due to cross pollination, and that's why hybrid varieties are expensive. But if these hybrids are made into apomixes, there is no segregation of characters in the hybrid progeny. Then the farmers can keep on using these hybrid seeds to raise new crops year after year, and they do not have to buy hybrid seeds every year. And because of this, active research is going on in many laboratories around the world to understand the genetics of apomixes and to transfer apomictic genes into hybrid varieties. So here we have completed the chapter. Thank you very much for watching, and don't forget to share and subscribe.